Okay, our final section here on biological molecules is nucleic acids. Last but certainly not least. So what are nucleic acids? Well, nucleic acids are DNA and RNA. That's how they fit into our bodies. So DNA, of course, is very important. You've heard about DNA. It contains the entire genome. It's all the information to make all the proteins in your body and then some. So basically all the information to make you. And we're going to be talking about how that works in detail in the DNA section, the genetics section. But so nucleic acids, that's the final biological molecule. So we got DNA, we got RNA. The basic monomer of nucleic acids are nucleotides. So we have to look at nucleotides. Remember, monomers are the building blocks, the small piece. So for lipids, it was kind of fatty acids were the monomers. For proteins, it's amino acids are the monomers. For carbohydrates, it would be glucose and its isomers, glucose, galactose, fructose would be the monomers, the building blocks. And for nucleic acids, it is nucleotides. Those are the building blocks, nucleotides. So let's look at what a nucleotide looks like. So for both DNA and RNA, you have nucleotides, okay? And the thing that differentiates DNA from RNA nucleotides is these 5-carbon sugar. So a nucleotide is basically composed of a phosphate group. Phosphate group is just a phosphorus atom with some oxygen atoms flanking it all around. It's got this 5-carbon sugar, or a pentose sugar. And the most important, or one of the most important bits, a nitrogenous base, which is over here. It's just something that's basic. It's a little structure with nitrogen that is basic. And the base will determine which nucleotide the nucleotide is. The pentose sugar determines whether the nucleotide belongs to RNA or DNA. RNA stands for ribose nucleic acid, and DNA stands for deoxyribose nucleic acid. And so the ribose is that pentose sugar, and whether that sugar has an oxygen or is deoxy, doesn't have that oxygen, determines whether the nucleotide belongs to RNA or belongs to DNA. Okay? So let's talk about these nitrogenous bases. The nitrogenous bases come in 2 families. There are purines and pyrimidines. Pyrimidines have 1 ring, purines have 2 rings. That's the basic difference between the two. And then what happens with these bases is, so we basically have 5 different nitrogenous bases that exist. There is adenine, thymine, uracil, guanine, and cytosine. You're only going to find thymine in DNA, and you're only going to find uracil in RNA. So thymine and uracil are basically the same but different. You're only going to find uracil in RNA. You're only going to find thymine in DNA. You cannot have uracil in the DNA molecule, and you cannot have a thymine nitrogenous base in an RNA molecule. Okay? And so the key to how information is stored in DNA, so genetic information is stored in DNA, is actually these nitrogenous bases, because what these nitrogenous bases do, they're essentially like letters. So you've got adenine A, guanine G, cytosine C, thymine T, uracil U, and what they do is they complementary base pair to each other. The nitrogenous bases can pair to each other with hydrogen bonds. Remember hydrogen bonds. So they hydrogen bond to each other. And so an adenine can hydrogen bond to a thymine or a uracil. A guanine hydrogen bonds to a cytosine. Okay, so it's called complementary base pairing. You may have heard DNA is described as a double helix. So DNA is a large molecule. DNA is a big, huge molecule. It stores all your genetic information. Every single one of your cells, with the exception of your sex cells and red blood cells and probably some other odds and ends. Basically, every single one of your cells contains the entire DNA genome. Contains all the information, and it is shaped as a double helix. Remember, we already saw helix in protein when proteins fold, the full helix. Well, here we have a double helix. A DNA molecule is formed into a double helix, and it's a double helix because there's two strands that are complementary base paired. So that's how the two strands stick together. So if we look. If we can find a good image, here we go. So this is a good image of a DNA, and it's been flattened out. It's not in its double helix format. But so basically what you have is you have these backbones, and the backbones are the sugar phosphate backbones. So each nucleotide here, the sugar and the phosphate, they're linked by the phosphate. These are going to be covalent bonds. To link these nucleotides together, you're going to have to do dehydration synthesis, which is going to use energy, and it's going to produce a water. And then across the strands, so these are all covalently tough bonded, the backbones that twist around covalently bonded, and then into the center are facing the nucleotide. The nitrogenous bases are facing in. And they are hydrogen bonded. So that's the weak bond. The hydrogen bond is the weak bond. And so what you have is you have a thymine to an adenine, a cytosine to a guanine, and basically that's what you get. And we'll talk about how that stores information, how that works later on in protein synthesis. So there's these hydrogen bonds that keep the two backbones together, that keep the two DNA molecules together. So that's for DNA. For RNA is single-stranded, so it's smaller. Here's a RNA molecule. So it'll also have this helix, the backbone. It'll have the same structure basically as DNA, except it's missing the other half. So it'll have the sugar phosphate backbone with the nitrogenous bases sticking out. The only base you're not going to find is thymine. You're going to have uracil instead of thymine. And now, so what you can do is you can unzip. You can break these hydrogen bonds in DNA, have the nitrogenous bases exposed out, and you can have a RNA molecule, nucleotides, come and complementary base pair. So if this is an adenine here in the DNA molecule, then a uracil RNA nucleotide will come along. If this is a cytosine, a guanine RNA nucleotide will come. Then you have an enzyme stitch these two together using dehydration synthesis, and you've made an RNA molecule that now can take that information out of the nucleus and into the cytoplasm, where it can perform protein synthesis and make a protein out of that. So RNA is usually used for, it's smaller, it will take pieces of DNA out of the nucleus. Also, RNA is used to make ribosomes, which are the site of protein synthesis. It's where proteins are made, or the peptide chains are made. So there'll be an RNA there. And there'll be 3 types of RNA involved in that, which is messenger RNA, that's bringing the genetic information out of the nucleus. Ribosomal RNA, which is part of the ribosome, the site where protein synthesis happens. And transfer RNA, which is an RNA molecule that brings the amino acid that is used to make the enzyme. And the details of that we'll talk about later down the road. And it's this complementary base pairing that makes all of those mechanisms possible. So RNA, DNA, and then the last bit of, so I talked, I said there's DNA and RNA. And so RNA is messenger, but there is a special kind of RNA nucleotide, a very special kind, and a very important one. You guys may have studied before, you've learned that mitochondria is the powerhouse of the cell. And inside mitochondria, sugar is broken down and oxidized, and electron chain, all this kind of stuff happens to sugars. And the final product is something called ATP. You may have heard of ATP. ATP is the currency, is like the energy currency of the cell. Where there's an ATP, you're going to have energy, and you're going to be able to carry out reactions. So remember when I was saying protein or dehydration synthesis takes energy? Well, the form of that energy is going to be ATP. You're going to need ATP to stitch monomers into polymers. And that ATP comes from in us it comes out of mitochondria. It also is made in the cytoplasm. Now, ATP is called adenine triphosphate. Adenine, which is one of those nitrogenous bases. Basically, what an ATP molecule is, the currency of energy in the cell, is a modified RNA nucleotide. It's a special kind of RNA nucleotide. It's very important. Look how important nucleic acid is, how important RNA is. RNA is used in every step of protein synthesis. There are 3 different types of RNA used to make a protein. Remember, I told you protein. We're proteins. We're made of protein. RNA is the energy currency that is required to make those, to assemble those proteins. And a modified version of RNA is our entire genome, because you could say that DNA is just a special kind of RNA where the ribose sugar has lost an oxygen. It's a deoxyribose sugar. Saying RNA, really important. RNA can occur spontaneously in nature too. You can have it in like asteroids and stuff. There'll be biological organic compounds where you'll have some kind of weird RNA. So ATP, let's just look at the structure. You need to be familiar with the structure of ATP. So basically we've got a nucleotide here. So if we're going to have a nucleotide, we're going to need a sugar. And since this is a modified RNA, so our sugar is going to be ribose. We've got a nitrogenous base. It's adenine triphosphate. So the nitrogenous base is adenine. And then we've got a phosphate group. And here is where things get funky, is instead of having one phosphate group, like we saw everywhere, like we saw in DNA and those RNA molecules, we have 1, 2, 3 phosphates linked together in a chain. And so that's basically sugar in your body. The ultimate goal of sugar in your body is to bang, 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 get disassembled and create enough energy in order to be able to stitch these phosphates onto a modified RNA. And why are these important? Because the bonds here, these last bonds, these phosphate bonds here, are called high energy bonds. They're these high energy covalent bonds, but they're weak covalent bonds. They're high energy, but they're pretty weak. They're still strong covalent bonds, but for covalent bonds, they're weak. And basically, I guess they're like under tension, or I don't know how to describe it. You know, this is all like atoms and stuff. So, but these are high energy bonds. And what happens is when these bonds break, when you cleave off a phosphate from ATP to make it ADP, so ATP is adenine triphosphate, 3 phosphate groups, into ADP, adenine diphosphate, 2 phosphate groups. When that bond breaks, it releases energy. And the cell can use that energy to carry out metabolism, to stitch things together, to carry out other chemical reactions. Now, interesting enough, maybe you guys knew this already, or you figured it out already, but I, for years, even after university, like 20 years, maybe 20 years, I always was like, yeah, ATP it turns into ADP and that releases energy. And then I was like, okay, yep, But I never thought about what form is that energy. Is it like electrical energy? And then I started thinking about it, and it's like, well, we're not electrical. Humans don't run on electricity, so it's not electrical energy. What kind of energy is it? Because it's always shown in diagrams. It's always actually shown as like electricity shooting out of the bond. It's a high energy bond, and when it breaks, it shoots out energy. But that's totally wrong. It's not electricity. what actually comes out is just heat. It's actually kinetic energy. That's what heat is. It's kinetic energy. So chemical reactions can happen in the presence of ATP being broken into ADP because it heats the area up. It increases kinetic energy and basically makes molecules, vibrate and slam into each other. It gives them like a kick to slam into each other and carry out chemical reactions. That's what's happening. That's what ATP does in your cells. So when you are running or when you are jogging or you are using a lot of, you're using a lot of energy, you are then taking a lot of ATP and turning it into ADP. At the same time, you're also creating more ATP. Your mitochondria are working. You're breaking down sugars. You're breaking down fats to make this ATP, to make energy, to make your muscles move, to carry out chemical reactions that make your muscles move. So you may have noticed, if you've ever gone jogging or done physical activity, you get really hot. Have you ever wondered why you get so hot? You're sweating, you're hot, your body is hot. Now, when you ask people on the street, why do you think you get hot? A lot of them say, well, because you're moving, and so the friction of your muscles creates heat. Sure, it creates a little bit of heat, but actually, what is creating the majority of that heat is ATP, that bond breaking, and a tiny, just think of these molecules. These are particles. We've never seen an ATP. Even with the most powerful electron microscope, this is minuscule thing, the tiny, microscopic, atomic sized molecules that bond is breaking, and it releases a tiny bit of heat that your body uses to move your muscles. And then so there's a little bit of extra heat, essentially. So when you are running, you are burning sugar, you're burning glucose, producing ATP, breaking ATP, creating more ATP, and all of those little all that little heat gets added up to heat your entire body, to heat you up. I think that's pretty cool. So to sum up, nucleic acids, very important. Obviously, nucleic acids are used for storing genetic information in DNA, transferring genetic information, moving it around in the cytoplasm out of the nucleus. So that'd be like RNA, mRNA. Used for protein synthesis, which is transferring the genetic energy is used for protein synthesis. So making proteins, using that information to make proteins. And nucleic acids in the form of ATP are used as energy by your cell to make chemical reactions happen in your body, metabolism happen in your body. So very important. And that is the end of Unit A. There you go. Thank you. See you for Unit B.